Right. Okay. Good morning. Everyone here? No? Yes? How's your break? Short. You already had a week break during the Eid, right? So how many weeks break you want? At least a month. So that you have to go back to school. Um. So welcome to the other final half of the semester. Okay, that's the reality of it after you have had your mid-semester break. Looking forward to finish the course. Uh, we still have three more major topics to cover. That is the plant growth regulators, the hormones that we are looking at today, and then the photosynthesis, and also the how the products of photosynthesis is transported, the concept of source and sink. Okay, and then usually, if according to your syllabus, we should be learning about one special topic. So last year I kind of combined that with the postgraduate class, but given that this semester you have a number of Mondays and Wednesdays holidays, I'm not sure whether that's possible or not. Well, we'll see. We'll see. Okay. Or do you want class ganti? Right. You got your notes? Yeah. Your notes always have the soft copy in the folder. Okay. If you're not happy with this thing, you can print singly on as you like. So let's learn about hormone. You got your notes? Got your notes. Lepas tu kau halau dia pergi duduk sana. Right, okay. If you do not like biology, this is going to be difficult. That's the truth about it. Because this is really talking about the process that happens in living organism. Because if you give hormone to your speaker, your pen, nothing gonna happen. Okay, but since living things, they are always ready to receive signals, to receive stimulus, meaning that they are dynamic, actively changing, actively interacting and responding to the environment, right? Even though it sounds like a bit much, but you should celebrate this as a common ground between you and plants, because you too have hormones, okay? And in fact, you have been learning hormones since your school time. So the concept of hormone should not be an alien topic for your comprehension, okay? Who have never learned hormone before? Do you have hormones? Are you hormonal? Do you know what hormonal means? I'm surprised the guy said yes. When somebody says that you are hormonal, that's not a good thing. That means you are irritable. You know, kau suka, what's the Malay word for that? I know there's a Malay word for that. Kau suka meroyan. That means you are hormonal. Are you hormonal? Yes. Okay. So plant there. Oops. What happened? So there are many synonyms, but they all mean the same. So plant growth regulators can be described as PGR, growth substance, plant growth hormones, or phytohormones. Yeah. But since hormones in plants are regularly used in the synthetic forms, that's why you can see that sometimes people don't use the word hormones for plants. They use regulator because they have specific intention to make certain things happen to the plant. And if that was the natural process, it wouldn't happen. Okay, so that's why the word hormone is used. Pretty much like our government like to use the word incentive because they do not like to use the word subsidy because subsidy sounds you are very spoiled, right? Therefore, they use the word incentive. But actually, the root of it kind of the same. Yeah, people still receive money for doing something, right? And it originated from the Greek hormone, which means to excite, meaning that originally everything in the ground state, nothing exciting happening, and then when hormone comes into the story, things get excited. And it is a natural chemical messenger transported, meaning that hormones not necessarily stay in one place. It moves from one location to other location to the targeted cells, right? And it is needed like vitamins and mineral only in minute concentration. Okay, you do not need um, hormones as much as you need carbohydrate. No, because this is to trigger. Okay, to trigger means the organs is already capable of doing that particular process. You just need to tell it to do so. Yeah, and if you poking the organ too much, that's not gonna yield any meaningful result because that might damage the organ. That's why it's only a very minute amount needed. And another terminology is the phytohormones. A phyto means plants, simply. So plant hormone, phytohormone, and it has been around since like 70, 80 years back. Why use different synonyms for this phytohormones, plant growth regulators? For phytohormones, when you see certain text or people use these, they are actually referring it to as the organic compound, okay? Naturally produced in plants. If you're talking about the other synonym, which is plant growth regulators, that could be synthetic, right? Yeah. So hormones, there is a natural connotation associated to it. You wouldn't use the word growth regulators for humans, right? Why do you hate properties? Oh, because my organs are producing growth regulators that make me do so. No, you use the word hormones. So there is a innate association with it. Hormones, natural. Growth regulators, synthetic or unnatural. Since both plants and humans are producing hormones, so what are the key differences? Because these are two different organisms after all. Unlike in animals, plant hormones, as it says there, not produced in specialized tissue or cells, meaning that plants do not have glands. You know glands? Glands. Do you have glands?
Yes.
What glands do you have? There are a number of glands. You got your pituitary gland, you got your adrenal gland, pineal gland, right? And all of these glands producing specific kind of hormones in human. But in plants, that is not the case. Pretty much any cells is capable to produce the hormones. Yeah, but usually it's rather located or regionalized within the plants, because why there is a difference in this? Why we are very targeted for that? Well, to start with, humans are mobile. We can move on and about. We have more interactions with our environment. Okay, for plants, it's pretty much sedentary, and the circulation system is not like human. Humans have heart to pump the blood and to circulate. Plants do not have that. So if they restrict the hormones only for certain cells, they are going to miss out on certain signal receptors events, because there is no special circulation to force it to go all the way to the end of the plant. Okay, it can reach there, but it's rather slow. It's not like human. You can pump the blood within your body within minutes. You know, if the blood starts from heart, coming back to heart, it will only take minutes. Okay, but for plants, that might take some time to happen, maybe hours or maybe days. So rather than waiting for the bodily fluid to reach the entire body, just produce the hormones wherever it's needed. So that's why plants have multiple places to produce the hormones. Yeah. There are many hormones in plants. So, but for the sake of your level, these are the hormone classes. Okay, there are many hormones. Let's say that there are 10,000 hormones. All these hormones belong more or less under these classes. 5 main classes: the auxin, cytokinin, ABA or abscisic acid, gibberellins, and the gas hormones such as the ethylene. Okay, and all of these in the form of fluid, liquid, except ethylene. Ethylene is the only one hormone in the form of gas. Do you have gaseous hormone? Yes or no? No? What about pheromones? You know pheromone? You know pheromones? Not the pheromone to attract the flies. No. Humans got pheromones too. You see, babies, when they first born, they don't really open eyes. Do you notice that? Or you have not seen babies before? But if you notice, the blind babies in the first weeks of the life, it likes to cry a lot, right? But when the mom is closer somehow, it stops crying. And the mom doesn't have to make a sound or anything, just stands next to it and then prepare the baby diaper stuff, and the baby will stop crying. That is one of the evidence the baby is actually sensing the mom, which is not involving any contact. Okay, so it's the mom releasing pheromones. Actually, you got pheromones. That's why some people, when they have crush to somebody, they like to stay next to the person. That's the pheromone. That's the pheromone talking. Okay, you don't have to touch the person. You just like the presence. You know, the presence. Sometimes you do like the smell. Maybe the guy or the girl is wearing expensive perfume. I'm not talking about the perfume now. It's just you just like next sit to the person. Okay, what if your head sits next to the person? Probably there's a pheromone. There's a evading pheromone. Yes, you see. Don't. Please go away from me. Okay. So all these hormones in humans as well, we can produce some kind of um, you can say it gaseous hormones, but I do not know how far scientists have studied about it. There are studies involving that. Okay, but for plants, surely ethylene is there. There are abundance of studies involving ethylene, and it's very fascinating, really. And from these five classes of hormone, they are further divided into two. Hormones that are regarded as growth promoters and inhibitors. Promoters meaning that they incite growth like elongation, expansion, proliferation, and so on. And these include auxins, cytokinins, gibberellins. And then you got the inhibitors. Inhibitors meaning that to stop, to make it less young, like senescing, or to completely stop it from acting in a usual manner. You see certain tissue in plants, especially the reproductive tissue, if ethylene is applied, you can kill the male tissue, and that is inhibiting. Okay? And this is why in agriculture, if you apply ethylene, you can change the sex of the plant, the flower. You see, the sex can be changed. Why? Not because the hormone doing surgical method to the plant. No, it prevents the growth and kill the male tissue of the flower, leaving only female. Right. So there are types of PGR, and one is called the promoters that you see, auxin, gibberellin. The other one, cytokinin. Okay. There are two groups, and one is they are promoting the growth, such as cell division, cell enlargement. I have mentioned this earlier. And why do plants need to have all these various hormones just to promote growth? Can't it be only just one hormone and then that's it? Because of this. Because plants want to be specific in either it wants a division, or does it want an enlargement, or is it want to target specific organs, flowering, fruiting, or seed formation, or vegetative growth. So it's not like human. Your red blood cells, can it enlarge? Your red blood cell, do you have red blood cell, erythrocytes? Can it enlarge, become bigger? Why? Why not? It doesn't have a cell wall. If it enlarges, it's going to pop. Right? Your liver cell, your heart cell, your myocardial cell, can it enlarge, expand, swell up? Definitely you're going to burst up the cell. Plants, because of the presence of cell wall, the cell can swell up, can enlarge, can double. So many things, dimensionally speaking, can happen to the cell. This is not possible for humans. Okay, and in fact, there are many neuron cells that has been around since your birth. It's pretty much stay that way. The cell in your eyes. I don't know whether this is true or not. They said that your eyeball is pretty much that size since you were born. They don't really enlarge. If they enlarge like your hand, your baby hand is like 1/10 of this hand now. If your eyeball becomes 10 times bigger, you know, instead of eye, you have what? Coconut. That will make you look very weird, alien-like, right? So not all cells in your body grow or expand proportionately. Okay, some stay that way since your birth. Okay, what else stay the same since your birth? It doesn't increase. It doesn't increase in number, meaning that it does not proliferate. That's the word proliferation. Or I forgot to mention that. The numbers stay the same from the day you were born. What? Hair. What? You were born, you got that much. By the age of 40, you got that much as well. What's that? Finger? Fingers not cells. Fingers is organ. Ears? Ear cells. Your ear do not get bigger. Actually, your ears, as you get older, it will grow longer. Your earlobe will go longer. If you don't trust me, look at the 100 years old person. You can see that the earlobe is actually kind of coming down.
The eyelids.
Eyelids. Cornea. They actually got replaced. Every 8 years, the entire atom in your body actually got replaced with a new one. You are not you. The egg cells. Aku tak tahu lah, aku tak telur. When you were born, I think the story is like something like this. When you first formed in the fetus, you got something like 2 million eggs. When you are about to be born, also it's got reduced to 200,000 eggs, and then when you are born, you are only left with, I think, around 300 eggs. And these are the eggs that are going to mature and then become your ovum. So he says the same, right? It's not suddenly at the age of 15 because you got excited, you go to concert, your eggs multiply. No, that's not what happening. No matter how much you got excited because you are seeing your favorite band, your eggs not going to get multiplied. It stays 300 from the beginning, and that's what you get. Kenapa kau gelak? Your eggs got multiplied. That's what you believe. No. Right. So humans can't do that. Right. The eggs do not get multiplied, but they do get mature in the corpus luteum, in the ovary. Okay. The guys I know, they cannot appreciate this. They don't have got your problem. It's okay. I understand. Yeah. That before your red days happen, you are like just joining a cult. Right? So if your friends, your girl space friends is unreasonable, cubalah memahami. They cannot control that. That's evolution. Right. Okay. Another type is the inhibitors. So we're going to have a look more on this. I have said the fundamental of it. It's to inhibit. Inhibiting is not necessarily a bad thing. Okay. Inhibit to do one process in order to allow another process to happen. Right. If you are not get inhibited, you are as tall as coconut tree now. Tell me, which supermarket is going to sell your clothes? So inhibition is not a bad thing after all. Okay, but we do not want it to happen before the time. Okay, we want it to happen at the right time so that the organism, including yourself, can function beautifully. Right. So let's talk about the first um, group of hormones, that is auxin. So it was first not discovered actually, but proposed by Charles Darwin. You know Charles Darwin?
The monkey.
The monkey guy. What? Yeah, yeah. The fathers of modern evolution. Yeah, right. Um, I during my intern, you got to do intern. The intern. I did like 2 weeks my intern work in Charles Darwin home in what's the name of that county? Kent. His home has got a home. Anak dia ramai, anak dia 10, so the home is so big. So he did a simple experiment. I'm going to show you, and he noticed that if you allow the seedling to germinate, to grow, when there is a light coming from one side, like in here, the coleoptile means the protective sheath of the plumule, will start to grow towards the light. When the tip is removed, it stays erect. When the tip is covered with something opaque, it's going to stay erect as well. But when the tip is covered with something transparent, it follows the control. When the only the base is covered, it will follow the control. So he started to postulated that there must be something, some kind of chemical or substance that get produced within the coleoptile region, the tip region. Okay, and then of course he died. I think when he died, he got buried, and he's now neighbor with Newton, the same place. You know Newton? Did you learn calculus? That's from Isaac Newton. They are neighbor now. Betul, kau tak percaya? Kau tak percaya? Kau cari benda ni. I think this is where they are buried. Bukan Abbey. Westminster Abbey. You know London got the clock, the Big Ben. Okay, next to it. Westminster Abbey, next to it. Belah aja. Very close. It's like here, and then you go to Dean's office. Right. Okay. So and then the other guy continued the experiment, and then between the coleoptile and the main body of the seedling, he separated with two kind of blocks: the gelatin block and the mica block. The gelatin is porous. It allows whatever compounds, if ever present in the tip, to go down. And when he used the gelatin block, the coleoptile is following the control. But when he used the mica block, mica is pretty much is a mineral, like a stone. No response whatsoever. So now they are confirmed there must be something in the coleoptile. It's not in any other regions that control this phototropism for the seedling to follow where the light is. Okay, and then when the time progresses, the scientists, so this is where you can appreciate the work of scientists. It's not the work of one guy, one scientist. It's a continuous work. This is why I keep stressing on a good scientific conduct. Because whatever your discovery, whatever your observation, it's going to be useful somehow down the line by other scientists. Okay, so the other scientists isolated, meaning extracted the compound from the coleoptile tip, and its name auxin, from the Greek. Auxin means to grow, and the form of the auxin. Remember, there are many of auxin that got extracted in the form of IAA, indoacetic acid. Generally produced by the growing apices of the stems and roots. See? Stems and roots. These are two completely opposing organs of the plant, end to end. One at the end of the shoot system, and then one at the end of the root system. You don't get this in humans. Okay, you don't get your pituitary is produced in your pituitary gland. Your pituitary got produced in your finger, in your hair. No, your hormones pretty much produced by a specialized gland. Your pituitary gland is not going to produce estrogen. What happens if your pituitary gland producing estrogen? What potentially will happen? Meaning that you get estrogen activated all the time, right? If estrogen get activated all the time, you are not able to grow properly. Okay, you will start to look funny. You'll feel funny, and more or less, you're not going to be human. Yeah, yeah, yeah. So is estrogen is a bad thing? No, estrogen is not a bad thing. It has to be around in the right time, in the right amount. Okay, that's why in human it's specialized. Right. So what? And then scientists wonder again, what's with the bending? I mean, like, if in the whole cylinder of the polyptile, is there any specific part of the cylinder is actually producing the auxin? Is it here? Is it here? Or is it here? So the scientists started to think about this because they hypothesized that in the cylinder region of a polyptile, there must be one region that is producing auxin because they believe that localized knowledge from the human. Because a human, you got the gland, right? So since polyptile do not have gland, there should be some regionalized production region for the auxin. So what this scientist did was he cut the auxin and he put it in the agar to transfer whatever auxin that is present in the polyptile into an agar block. So this agar block now contains auxin, and then this auxin is placed on the decapitated polyptile A and polyptile B, in a rather asymmetric placement. It's not completely on top of the cut head, but just on the edge, in this manner. And they see that when it's placed this way, the bending is occurring on the shorter side where auxin is not placed. So now the theory is confirmed. Auxin is actually present when the plant is bending in certain way. The longest curvature is where auxin has to be the most. Right. So of course, the experiment got further proven, and they cut it. They put it on either side now. No matter which side it's putting, whether it's this side or this side, the curvature will be on the opposite direction. If the auxin is placed on this side, on this edge, the curvature will go the opposite way. If it's placed on this side, the curvature will go on the opposite way as well. So what's with that? Why? Why won't it form the curving according where the auxin is placed? You'll see in a bit. So it turns out that later it was found that the auxin is actually shying away from the light. Okay. So when a polyptile like this is facing the light, auxin actually shying away or moving away from the light. When this happens, the the darker side of the coloptile will be concentrated with auxin. And this is why the longer curvature happens on the darker side, while the side that is facing the light will curvature or curving the least. And that's why you got the bending towards the light source. And this is the explanation for why phototropism is happening. Why auxin is shying away from the light? And why is that? This is the nature of auxin. Okay, there are many compounds that are actually sensitive to light and temperature. For example, vitamin C. You see, if you cook your vegetable and fruits for too long, above 70 degree, that's going to destroy vitamin C. That's why it's not healthy to eat fried fruit all the time, baked, roasted, satay, that kind of stuff, because definitely no vitamin C in there. Why do you think after you have eaten satay, people give you all the onions, the cucumber and so on? The wisdom is there because there's no vitamin C left in your satay. So you got replenished for your antioxidant by eating all these salad cubes. Burning your meat is not a healthy way to eat it, but people eat it because it tastes so damn good, right? So to compensate, to neutralize the free radicals that's happening in your body, eat more salad. All right. Auxin, there are two types of auxin. There are the native auxin, meaning that you can find this auxin naturally in plants. You can have the synthetic form produced in the factory, but in plants we got this. We got the IAA, indole acetic acid. I do expect you to know this, okay? So at your level, when people want to ask you which auxin are you using, you need to know what kind. Is it IA? Is it IBA? Is it NAA? Okay, right. And then you got the IBA, butyric acid, and then phenylacetic acid, PA. And then you got the synthetic auxin. Okay. These synthetic auxin, they have other functions than stimulate growth in plants. They can be used as herbicides. Have you taken your weed science?
No.
In weed science, later you will learn there are many types of herbicides. And some of these herbicides, actually, they are auxin, the synthetic auxin. Okay, you got 2,4-D, alpha NAA, dicamba, amprodon, and picloram. There are many types. So, and these are the properties of the auxin. So, the natural auxin, the IA, is broken down in sunlight, and this is why it's shying away. If vitamin C get deactivated with high temperature, auxin get broken down with sunlight, the light. Okay, and these are the commonly used auxin in horticulture. You got IBA, NAA, naphthalene, acetic acid, and also 2,4-D. 2,4-dichlorophenoacetic acid, phenol C, acetic acid. How are you going to remember that? Use whatever power left in you. So where auxin got produced? In the shoot tips, root tips, young expanding leaf like the plumule, and also the young seeds. So natural auxin, and therefore can be regarded as phytohormones because it's natural. Remember, hormone is associated with natural. And the synthetic auxin can be very cheap because they can be produced, manufactured synthetically. Even though they are synthetic, they still incite similar response to natural auxin, right? And however, synthetic auxin is not called phytohormones. This is when you start to use PGR, plant growth regulators. Okay. And these are the compounds. I just want you to keep on seeing these names because you need to commit these names now to memory. Right. Oops. So the function of auxin, there are many functions of auxin. It's not like human hormone. Estrogen is for all the female stuff. No, auxin pretty much doing various function. You got phototropism that you have seen in the Darwin's and Friends experiment. Gravitotropism, phytotaxis, senescence, fluorinization, apical dominance. But when it comes to cell elongation, it can be the opposite. When you have a lot of auxin in a root organ, the effect is inhibiting. Okay, remember, okay, auxin can be inhibiting in one exception in the root here. You will see in a bit why. If auxin is a lot in the shoot part, in the shoot organ, that promote growth. But auxin, when there's a lot of it in the root region, that actually limits the growth. And this actually limitation, remember, not always a bad thing. It can be a useful thing for the plant, right? Do you know phytotaxis? Have you seen that one before? No? Have you taken your botany? You didn't learn phytotaxy? No?
Regular arrangement.
Apa arrangement?
Arrangement of leaf.
Okay, how many arrangement of leaves? 8? What's this? Alternate. Whirl. Hurl. Hurl. Ni? Rosette. Rosette. Satu lagi. If your rosette suddenly have bolting, and then you got your leaf here, this is called colin. Colin leaf. Okay. Janganlah pelik. I teach botany too. But not our faculty. Other faculty. Right. Okay. Let's move on. The way auxin moves in plant. This is important. It's called polar auxin transport. Why is it polar? So there is a concept in plant. It's called the root shoot junction. So in order to understand this concept, polar auxin. By the way, you do not understand what is meant by polar? Not polar express. Polar means opposite, end to end, south, north, east, west. Like the magnet. Magnet will attach to each other when it's the same pole or different poles. Different pole, right? So polar, it means the opposite. So end to end, opposite. Sorry, I want to write east and west. North, west, south, east. Okay, and then you learn about the new terminologies here. I think it should be mentioned there. Acropetal, basipetal. Have you seen this word before? Acropetal means towards apex. Basipetals mean to base. Okay, right. So auxin, it will always go to the base to the apex somehow, right? I'll show why. When you look at this plant this way, you need to understand now, the plant is divided into two body regions, the shoot region and the root region. And the base of it is called the root shoot junction. And this is the important bit. Okay. The moment you go beyond this root shoot base junction, the orientation of the plant gets reversed. Okay. This is shoot apical meristem. Okay. So when the movement involved from the shoot apical meristem to the base, the junction base, it is called basipetal. So that's normal. If the transport of the chemical starting from the junction base to the shoot apical meristem, that's called acropetal. That's normal as well. But when you go to the root, this has now become the apex. Okay, so the movement of chemical from the apex of the root, which is at the bottom of the soil, all the way to the root shoot junction, this is called basipetal, towards the base. However, for the acropetal, from the base going down to the root tip, it's called acropetal, towards the apex. Okay, so this concept is very important, and do not get mixed up between the two. Okay, and the auxin, it will move like in here. This is the middle region of the seedlings, and then this is the root shoot junction. When this is transferred to a block, you will see that the auxin will go from up to down. Okay, because for this seedlings, the auxin is primarily produced in here. When you turn it, the transport is not happening. Okay, because auxin is actually kind of tied to gravity. Okay, if it was not for the gravity, even after you have turned, originally is A top, B down, it should be following from here to here, but that's not what happening. Okay, so due to the gravity, you'll see in a bit why gravity plays an important role in here. So your auxin can actually move from the bottom of the soil all the way up, but actually not all the way to this base. It's just in this region, but that movement is still considered as acropetal. Right? Why? Why the auxin from the tip of the root cannot go all the way up here? Because of the gravity. Okay, it just can't. From the top to down, from this shoot region to down here, that's fine. Okay, you are following gravity anyway, and you'll see in a bit. Right. So this you should be get familiar now. This is from the candle experiment. You have your auxin pretty much concentrated in the darker region. So remember this diagram for the shoot region. Now let's see the opposite teaching now, which says auxin will limit growth in the root. Okay, so here's the region. This is not shoot, okay? This is root, the tip of the root. When the plant is placed in the vertical position, the auxin that is originating from the top, from the shoot region, will come down through the stele. Stele is like the core of the root, and it will start to deposit in the tip of the root. Okay, that is one thing. The second thing is, you see this thing? This is statolith. Statolith is actually kind of like small stones in the plant. It's actually like a concentrated amyloplast or something. You got this as well. Where do you got this? However, in human, it's not called statolith. It's called otolith. You got this in your ears. You know your ears got stones? Yeah. You got stones actually in your ears. That's why your gegendang telinga, you learn to control your balance. That's why your body know whether it is to the right, to the left, to the front, terbalik. Because of this otolith in your inner ears. This otolith has got all the 3D structure. It will touch accordingly to the hairs in your ears and the movement of the fluid. So that kind of tell the gyroscope in your head what's your bearing right now. If you got disturbed in your otolith, that's why some people cannot ride bicycle. You know, some people cannot ride bicycle? Yeah. It's not because of Sophia sometimes, it's just they cannot balance themselves very well. Right. So in plant, you have statolith. Not for balance, but for gravitropism. Okay. Whether to ensure that the root is following gravity or otherwise. So when the plant is in vertical, you already have your auxin concentrated in the tip, which is because the auxin originated from the shoot. The statolith also concentrated in the tip of the root. Okay. So when this happens, the auxin that is produced locally in the root will start to produce equally on either side. Okay. So auxin has this tendency to be far away from the statolith. Whenever statolith is there, auxin kind of like, I don't like to be there. I just want to be shy away from it. Remember in the shoot, the auxin is shying away from the light. This is for the shoot. For the root, auxin is shying away from the statolith, the batu. You can think of auxin dislike batu. Right? Why? I do not know whether people have done the study about it or not, because the batu is not really a batu, it's actually a starch that concentrated into a deposit. Right? Okay. So when that happens, the auxin will go up on this lateral side, and then this side will undergo elongation. So the elongation of the root will go straight down. What happens when the plant is placed on the side rather than this way? It's placing lying down. So when that happens, number one, you will start to have your otolith concentrated on the down region, the bottom region of the root tip. When this happens, you will have less auxins present in this region. When there is a less auxin present in this region will undergo limited growth. What happened on the upper side of the root? This is not affected by the statolith at all. So this region will have lots of IAA, the auxin, plus also the IAA that comes from the top. So this region will grow the most. So the concept is pretty much like this, the light, but now in the presence of statolith. That's why you get your positive gravitropism. The root will grow towards the gravity. Why? Because statolith is heavy and dense. It will deposit where gravity is the most. Okay, got it? Right. Right. How auxin elongate the cell? So on the cell surface, the plasma membrane, there is a special protein that is called hydrogen pump. Okay, and the function of hydrogen pump is one, to pump hydrogen. In the outer region here, when auxin is present, it's going to activate the hydrogen pump to pump more hydrogen into the vicinity of the cell wall. When more and more cell wall is filled in with hydrogen, what is going to happen now? It's going to become acidic. More hydrogen, higher acidity. That as simple as that. That's why it's called pH, potential of hydrogen. When cell wall become acidic due to the presence of hydrogen ions in abundance, the cell wall will become softened and loosened. If you zooming out, you can see that the cellulose fibers in the cell wall will start to disintegrate and loosening up. Rather than they look too crowded, now they become more relaxed. Why? Because acidity. Right. That's why acidity is also the weathering agent when you learn the soil science. Because it can break things apart. Your stomach, what's the pH? Do you have stomach? What's the pH in your tummy? Is it alkaline or is it basic? Why is it acid? To digest your food, to break down your food, to activate the protein. If your stomach is not acidic, how is it going to break down the food so that your body can absorb to nourish yourself? Right. So breaking things apart is the specialty of pH, which is lots of hydrogen. And when this happens, the cell will soften and it can elongate. Why? Because remember, the vacuole that is filled in water is always pushing. You got your water potential. Remember you learned water potential before this? So there's also always a pressure that is pressing against the cell wall. Now that the cell wall is soft, it's good. When I press, it goes that way. Before this, when it's pressing, nothing much can move. Yeah. Can this happen in human? No. Human doesn't. To start with, humans do not have cell wall. So what happens when you go to your friend and you spray IBA to him? Will he grow? Macam you feel pity. You too short for your age lah. Come. Let's grow some more. Can your friend grow? Why? There's no receptor for auxin. Yeah. He's not going to grow, but he's going to get very irritated. Right. Okay. So yeah, that's how it works. Right. And finally, another thing is when it comes to growing, auxin. I don't want to go too much into this. This is actually a bit advanced, but it's enough to say auxin controls gene expression. The way it works is you have your auxin, then your auxin latch on the receptors that is always present on the surface of the plasma membrane. And when this happens, the receptors will cause a signal transduction, meaning that it will activate another compound in the cell. And then that compound will activate another guy will activate another guy, and eventually that signal arrive in the nucleus. When the nucleus got the signal, it will start the transcribing. You know, the chromosome will relax, the helicase will open the chromosome, and then it can transcribe, copy in, and then you will get your mRNA. mRNA will be translated into protein. And you got your protein. Right. And look at here, the auxin is not produced within the cell. The auxin is coming from somewhere else. To show you that the effects of auxin can be throughout the plant. It can move according to the polar transport concept. to the either end. Right. And additional response to auxin in addition to cell elongation, expansion, and proliferation. There's apical dominance, parthenocopy, and herbicide. Apical dominance. Oh, this is a favorite for all the gardeners. You have your shrub growing. If you allow it to grow, it will go up looking slender. That's not very interesting. Then the decay will come and start to chop off the head. The moment the plant gets chopped off the head, it will start to become bushier. Right? And that is the effect of auxin. Why? It's called apical dominance. Whenever the plant has the apical or the apex present, all the lateral buds, meaning that the buds that is present in the axillary. So you got your plant. This is your apex and then you got your branch, right? Then you got this. This is actually a bud, the leaf bud, but it stays dormant. It's not growing. Why it's not growing? Because of this phenomenon called apical dominance. But the moment you chop off the head, all these axillary buds will start to grow. There's nothing stopping it now. The reason is, when there is an apical dominance, the sugar that is produced in the side leaf is not going to feed the apical bud, but will go straight top, feeding the shoot apex. The moment the top is cut, the sugar from the side leaves will start to feed the apical bud as well. When this happens, that's why you get the growth. And allow it to continue for another 1 or 2 weeks. That's why your plant get bushier. Yep. And this is why in horticulture and also in forest management, pruning, trimming is very important. Okay? Because when you have more shoots, you will have more buds. When you have more buds, you will have more flowers. When you have more flowers, you will have more fruits. When you have more fruits, what will happen? You become richer. Jual lah buah tu. Right. So it's not a good practice if you only leave your plant that way. Okay. But of course, there is the art of pruning and trimming. You don't want to cut too much until your plant is stressed. Okay, you keep on cutting, it hasn't got enough energy to regenerate. Surely it's going to die, right? So this needs to be done accordingly. And then second thing is parthenocopy. Parthenocopy is the formation of the fruits without fertilization. So if you learn from your botany, you know that upon pollination, meaning that the pollen has landed on the stigma, the pollen will start to grow pollen tube, finding all the way down and then causing the double fertilization. Did you learn this? Belajar tak? Double fertilization. And this will give rise to the fruit. What one was an ovule will become seeds, ovary will become the fruits. That's why the seeds is always within the fruits, because ovules is in the encasing of the ovary, right? However, that requires fertilization. Mommy and daddy needs to meet together. Then you have your fruit. With the presence of auxin, when you only have your female flower, you have your ovary, right? You have your ovary, you have your ovules. Your ovules is not fertilized because there's no daddy coming in. But you still have the encasing, the cases, the vessel, the ovary. And this ovary is actually responsive to auxin. When farmers spray this ovary with auxin, this ovary, even without fertilization, they can continue the growth. They can continue the growth becomes the regular fruit. However, this is going to be seedless because no fertilization that took place. Unpollinated, usually this is what you get. The flowers just to wither, wilting away. Now you spray with auxin, these flowers continue to grow. What continue to grow? The ovary. Because the function of auxin is to promote growth. Very easy. Increase the number of cells and enlarge the cells. Okay. Why this is possible? Because the ovary has all the information that it needs to become the fruit, except seeds. Seeds, you need the switch from the daddy. So it's not possible to produce the seeds. Okay, which is good. This is why you got your seedless fruit. Look at your banana. Is there any seeds in the middle? There's seed but not fertile. Can you taste the seed?
No.
That's not even seed actually. That's just a remnant of the unfertilized daddy. Right. Okay. Right. And finally, auxin in the use of it in the industry use. There can be a concept of too much of good things. Yeah. In the past, people use auxin to win in war. Okay. It's called Agent Orange in Vietnam War. That's the story here. Kau baca lah. The enemy, when they come over Vietnam, Vietnam pretty much like us. Pretty lush jungle, tropical forest, right? So when the enemy come from the top, can they see the soldiers hiding, concealing themselves within the forest? They can't do that, right? So they spray the forest with this concentrated two kind of auxin, 2,4-D and 2,4,5-D. This thing caused the effect of defoliation during the Vietnam War. When this happened, why is it called orange?
Was orange and
What orange in color?
brown. They tell the foliage orange.
Yeah. The originally the whole forest looked green. Before the leaves fall down, it turns to orange first. It undergo chlorosis. The chlorophyll breaks down. When chlorophyll breaks down, what's left is the pigments, other pigments in the leaf. And these pigments are usually carotenoids. What's the color of carotenoid? Orange. That's why the whole forest now looks orange. And then they come and they bring the fan or the leaf go down. And they see, tu pak tembak mati. Dah tak boleh menyorok. Right. But people don't use that now. We use that to control the. There are many products. Okay, you can see in the active ingredient whether it's either 2,4-D or 2,4,5-D. Right. Right. Okay, that's all for auxin. What time now? Baru satu hormon. How many you got?
5, 4.
What week now? It should be week 9. You see, your class I have not done with you self-learning. And all the time I come and teach you. According to your study plan, nanti diorang bising. You need to undergo some form of self-learning, guided self-learning. Okay, since aku pun tengah sibuk, let's put it this way. I'll come and teach you again next week, but if you found the video letter, can you give to them from last year? You can use the video to assist you while learning the other hormones gibberellins, cytokinins, and ethylene. And I'll provide with you the question to follow as well. Okay, so that's one. But bear in mind, your notes now is actually different from last year, especially in the ethylene section. I've changed the ethylene and it looks a bit more updated now. Right. Um, for your in-class activity, you need to do one presentation. Yay! Yes. Yay! Yay! I'll, I think we can use this as presentation. So ini semua you need to baca. Don't worry, nanti I will recap again with you. I'm talking about this. The other lesser known hormones, but they are equally important. So what you should be learning while with me is this: auxins, gibberellins, cytokinins, abscisic, ethylene. So that's covered, right? Now there are 1, 2, 3, 4 that you shouldn't be learning too much, but you should know the existence of it, the history of it, and the function of it in plants. So I'm thinking to turn this into your presentation. It should be a short one, 10 minutes stop. And this is going to be a group work. So why not today you decide which group, which what? You got salicylate or salicylic acid, jasmonate or jasmonic acid, brassinospira, and trigalactone. Do you want to decide or you want to poll vote? Poll. Withdraw. Can you do? Remember, okay, it's not like I don't want to teach you gibberellin semua. You macam ni, you need to understand there is a wisdom why our education system make it in such a way. Later on, if you are continuing your study, pursuing your study, you go to university in other worlds, you will see that the lecturer is actually not teaching everything. You are not going to be spoon fed. The material is there. When you come to the lecture or you have your tutorial time, that's actually for discussion and to revalidate your knowledge, doing exercise and so on. So I use this small bit of hormone story to train you with the self-learning, guided self-learning, okay? Right. So when do you want to do the presentation? Wednesday. Nak ke? Boleh je. Next week. I forgot to bring my calendar. What do we have next week? I don't bring my calendar. Isnin? I'm thinking Isnin to turn it into lab for the hormone experiment. Have you created the vote? Give.
Wednesday is Saturday, right?
So you're celebrating Buddha birthday? Oh, he's celebrating.
You Buddha public holiday.
Yelah, sebab kau selangor kan, kena lah public holiday. You see, it's become challenging with me untuk to maneuver your class. Hai. Okay, while doing this, have you submitted anything before your study break? Because I did not receive anything.
I already submitted the report in the lab.
Where? I don't receive anything. Did you collect anything? Have you give anything? No?
In the right tree. I took a picture, but I didn't.
Did you notify in the group so that the lab assistant know?
No, I'm so sorry.
I need you to submit now so that I can evaluate and correct if there's something wrong with your lab.
In the link that.
Nanti, can you check? It's in what tray? Red tray.
Red tray.
Red tray.
Yeah.
Red or red?
Red.
Right.
So that's one group. What happens to other group?
Simply next Monday. Because it was
Let's put it this way. Because next Wednesday is holiday. If we start something this week, it's going to be difficult because we need to see after 1 week, right? So I'm thinking we push your lab for this Wednesday to next Monday. So next Monday we'll do the lab. All right. So what happened to this Wednesday? Please submit your work. Okay. Kau nak markah tak nak? Alright. Please submit your work. Okay. Lepas tu, siapa yang dapat? Ambil. Cepatlah. Boleh tak bertindak cergas-cergas sikit?
just minutes.
So who get what? So Watson, crop top, apa? BCG? Apa? BBG? BBG? The other one? Cetaceous. Okay, what you got, Watson? What? Watson, what you got? What? Jasmonate. So jasmonic acid. Crop top? Huh? Salicylate. Salicylic acid. BBG? Striga lactans. Prasinosteroid. If you refer to the notes, it's actually a small story in there. I didn't remove any story, but since this is your presentation, go find something more lah. Right? Tell to the world as if nobody knows about this thing. Okay, what does it do? How does it work? What plus the highlight of the story? Some example. Don't go crazy. 10 minutes most. If you want to present only one slide, that's acceptable as well. Okay, boleh. So when do you want to do the presentation? Next Monday we need to do the lab. Sebab this Wednesday, I'll give you time to do the submission. Next. I need to off my calendar. One moment. When is the Buddha birthday?
25th.
May
25th.
25th? 22nd. Let's target on the 29th. Okay, 29. Because you only have 2 experiments left. The the auxin and the GA. One is for the rooting, one is for the seeds germination using hormones. Yep. Let's target on 29. Okay. Do you listen? When is the presentation? Do we have anything in 29? Damn it. 啊 ke macam ni 28 30 ke 29. No, I'm not ambil cuti. Korang pun kena cuti. Tak boleh. We shouldn't be around. So 28 30. Ataupun 30 31. 30 tak, sebenarnya Wednesday tu boleh, tapi petang. Engkau akan bagi talk. Discuss balik lah dengan dia orang. Okay, alright. Okay, let's recap what you need to do. You do your self-learning for gibberellin, cytokinin, abscisic acid, and ethylene. So that's 4. Okay, and later, if I remember, I'll provide the question to assess your understanding. That's one. Secondly, you do your presentation for the groups. Semua ni, Watson semua ada tu kan? Jasmonic acid, salicylic acid, brasinosterol, and strigalactone. Okay, and third, this Wednesday, please submit your whatever yang tak submit lagi. Okay, because you only have 2 more experiments to go. Okay, submit so that if your lab report is not okay, there's some time for you to do the correction. Or you have the plan not to get full marks, then I'm fine. Okay, so boleh submit this Wednesday. You know what? Let's make it. Nanti I'll ask the lab assistant to designate the proper tray so that nothing is missing. Until the end of this week, I'll come back on Friday to collect it. Okay. So on Wednesday, if you don't want to come but you want to have discussion under the bed with your friends, do it. Okay. Alright. Boleh. So next week, we do the lab on Monday. Okay. Right. Okay. So I think that's all for today. The introduction to PGR, phytohormones, and the auxins. Any question? Any objection?
No.
Any regret?
No.
Maybe. Kamu suruh kau tak buat awal. Apa? Kau tanya? Tak nak sudah. Why the class look lesser people today? People not in the mood to come? I feel like it looks lesser today. Lepas tu, why? What happened to your friends? Didn't get bus to come back? Yeah. I just want to see. Watson, raise your hand. Watson, raise your hand. 3 orang je. Okay, 4. Apa tu? Crop top. 3, 4, 5, 6. Okay. So that's what 1 or 2 missing? No? One. What happened? Don't know ke don't care? Okay. BCG lagi. BC. Raise your hand. Okay, everybody present?
Yes.
Okay. Cetaceous.
1.
4. How many of you in the group? There's only 4.
6. 1, 2, 3.
1, 2, 4. Okay. 1.
Harris.
Okay, what happened?
What is I don't know what they call
It's in your
program.
Pergi main bowling? I'm not against college punya program ni, but you know how program always hantar surat dekat lecturer untuk minta exemption daripada kelas. Can I send surat to them to minta exemption from their activity? I think I should do that, right? Yeah. Macam mana lah kita nak buat aktiviti luar kalau you every week ada je yang tak datang. See? Actually kan every time ada your class tak pernah full. I always get letters coming to my email, pergi sana, pergi sini, kan, gigi putus, macam-macam. This always happen. So, macam mana? I don't blame the tooth, but you know, during my undergrad, I have interesting fact about my undergrad. So I attended 37 classes and farm practices. 37. I was the class monitor for 25. Without volunteer, tak volunteer, never volunteer. Tak tahu kenapa. I turn it into my game actually. This semester I sit there, kena jadi class monitor. This semester I sit kat belakang sekali, kena jadi juga. Sit kat depan, kena jadi juga. Wherever I sit, tetap kena jadi. So there was one semester I kena masuk lambat dalam kelas sebab tak nak kena pilih. Lepas tu masuk terus jadi juga. So 25 out of 37 class monitors. Lepas tu attendance 100%. Can you imagine all the years to 100%? I should clap myself lah. Kalau siapa nak bagi aku award kan? Do you think I deserve award masa student? But I didn't get it. Kenapa? I actually dikira mengharumkan nama UPM juga masa tu sebab I did my internship dekat luar. People thought I was going to fail. So kenapa tak dapat? Apa intern aku memang cemerlang lah, macam kau. So my attendance was full. I was the class monitor for 25 classes. Lepas tu I finished my internship in England. I got scholarship from macam-macam lah. And then habis degree like you, I got another scholarship for my PhD. Tak sempat combo lagi ya. I got it. Tapi lepas tu on the convocation day, actually kan masa tu semua lecturer thought they thought I was going to win that, apa benda dia panggil benda tu? Chancellor Award benda semua. So you got about RM4,000 masa tu. Ke RM5,000. About that much lah. But I didn't win it. Not that I care. Sebab 2 days before that, I went to the palace to receive 100,000. Yalah, scholarship aku. I got scholarship to do my PhD. So my convocation was on Saturday. I went to the palace on Wednesday. So disebabkan Wednesday tu dah filthy rich. So Saturday tak ada apa pun, tak rasa apa sangat lah. Tapi when I think about it kan, thinking about our system, why didn't the excellent student got the deserving award?
It's always like that.
So sebab apa? Sebab I didn't apply. Tu je sebab dia. Padahal kan my tick all the box tu satu tick semua benda, and I got more than any history student UPM pernah dapat. So your lecturer is actually one of the best student fakulti ni pernah produce. But I didn't get it. So my friends semua of course lah rasa tak puas hati. 4,000 kot kau tak nak bagi kat kita orang. At that time I didn't feel anything, but after dah sekarang kan bila thinking balik benda tu, not regret but thinking about our system yang kind of unfair kepada deserving people. Kenapa? You know all my story sebab my story masuk paper berita kot masa tu. And then that story got circulated the whole UPM. But I still didn't win. Sebab tak apply. If you want to give to the deserving individual, kenapa depends on apply tak apply? Right. So the moral of the story is you are going to be underappreciated the whole life. Tambah pula kau dapat anak dahaka nanti lagi underappreciated. Right. So is that a bad day of mine? No, it's not really a bad thing. I didn't look at 4,000 tu banyak pun masa tu. Sebab because I just received like 20,000 pound. And orang tengah sibuk with the ceremony kan. I'm thinking kejap lagi dalam bag nak masuk apa? Because the following day I flew back pergi to do my study. So it's a very busy day. So kind of macam dah tak kisah dah all the convocation and stuff.
Kira doktor tu master lah, tu tak degree university.
I applied master, that university tak bagi.
Oh, then straight away to PhD.
Apply. I sent for master. Dia panggil benda tu MPhil, Master of Philosophy. But that university, when they send the offer letter to UPM, they send offer letter PhD. Kind of like risky. If you finish your first year with flying colors, you get to continue your PhD. If you don't finish your first year in a decent manner, you'll be sent straight back home. So dia kan macam kejam lah. So kira macam aku berani lah juga kan. Do you kau ingat senang benda tu? Ni macam ni, korang habis belajar kan? Habis belajar the final semester kat UPM, lepas tu straight pergi kelas pagi esok semua orang putih dah. Buku tebal ni ada 4, kena tengok baca. Yeah, that's not very pleasant actually. Don't do that lah. Sebab tu sekarang ni dia dah tak bagi dah yang terus jump pergi PhD ni. Sebab dia punya failure rate tu almost 100%. Yeah. So, is that a bad experience? No. Tak adalah bad experience. It's just sebab aku kan rajin. I kind of sedar diri. Okay, alright, yeah. So if you don't get awarded, sometimes if you know you are deserving but you don't get it, it's okay. Things will come in other forms, right? Kenapa aku cakap benda ni tak tahulah. Tak apalah. Kenapa? Kau ingat aku pusing sangat? You know Asasi? I was the first batch. Interesting tak? Asasi dulu tak ada syllabus. When you masuk kan, dia guna syllabus first year, jabatan biologi, jabatan fizik, jabatan kimia. So you join tutorial dengan budak degree, walaupun you are actually Asasi. First exam, hampir ada 30 orang kan, 29 nak dekat fail. Fail pun lagi banyak lah. Even my mark pun borderline nak masuk ke C. Susah. So lecturer dulu sebenarnya kejam. Kalau sekarang ni lecturer dah baik. Ya. Dulu mana ada ni sangat. We use the OHP. You know OHP? Overhead projector. The instead of scribbling on the screen, the lecturer scribbling on the projector, then it's projected on the screen. Masa tu mana ada WhatsApp, mana ada Facebook. 2005. Kau kat mana? 2005. Where were you?
In my mom.
Kindergarten? 2005 you were kindergarten? Alright, okay. So yeah, I think that's all for today. Just a small story. Tuni. So please do your assignment accordingly. If you need time to do it on this Wednesday, do it. Because I'm a bit busy as well. If I kind of push it, none of us going to benefit from it. Okay? So do that, submit when? Friday. Friday. Maksudnya when I come 5 PM hari Friday, kalau kau punya assignment tak ada, memang nak kena lah. Okay, boleh ya? Friday. Wait. Please work nicely in a group. Jangan bagi seorang buat kerja semua. When are you going to learn? Right, okay. I'll see you later. Bye.